diarrhea. Okay, what about C. diff? The first major paper decreasing C. diff, randomized double-blind placebo-controlled trial, British Journal of Medicine, British Medical Journal, 135, C. diff 0 versus 9 at 53. That was the first big prospective trial. Look at this, the Annals of Internal Medicine. I know the medical residents carry this around in their pockets, best one, best journal. Surgeons, we don't read the journals, we look at the pictures. But 4,800 patients, basically 4,000 patients. What do they find here? 66% decrease. The journal residents carry this around in their pockets, best one, best journal. Surgeons, we don't read the journals, we look at the pictures. But 4,800 patients, basically 4,000 patients. What do they find here? 66% decrease in C. diff. Okay. Here is a big article just published December 2017, C. diff from 1.6 from 3.9 incidence. 6,000 patient prospective trial, 19 published a meta-analysis. The question was here, if you start probiotics the day you start antibiotics, you do not get C. diff. If you wait 3 days, which is what this study showed, timing is key. Okay, but what about this one? Look at this, another study. Decrease 60% risk. The more bacteria you have, the more probiotics you have, the better chance you have. This is 6,000 patients publishing in this big journal, different group. So we've got multiple patients. Do they work? Does it work? This is where I work here, the big hospital in Northwest, the big teaching hospital. We went from 2.2 incidents in the ICU, people got antibiotics, down to 0.7 by just instituting a probiotic protocol. Cost us about 61 cents a day. Remember, one episode of C. diff sepsis, 100 grand. So you can buy a lot of probiotics. But remember, you can't just use any probiotic. This is a nice study done in England where they said, okay, let's see if we can give yogurt and prevent this. Okay? So they took the most common yogurt sold in England. They then screened 17,000 people at 3,000 met criteria, and they basically showed C. diff, no change. Because remember, it's a strain-specific, species-specific, so you got to have the right species to do the effect. And I could go on and on about all these studies, sepsis, C. diff, ventilator-associated pneumonia, et cetera. But again, I won't bore you with that. A little bit on prebiotics. Remember, prebiotics is a substrate for the bacteria to produce. Prebiotics are, these are usually soluble fibers. Probably the ultimate prebiotic is breast milk. There's a tremendous amount of prebiotic in breast milk. 15% of the carbohydrate in breast milk cannot be absorbed by the baby. You think about that, evolutionarily, why on earth would we evolve to not let the baby absorb this nutrient produced by the mother? That nutrient is specifically not absorbed because it wants to make the right bacteria in the colon, to give substrate to produce increased lactobacillus, increased bifidobacterium. So Mother Nature is very smart, okay? It makes it that way. The number one killer, I do a lot of work in Laos, the number one killer in the first 6 months of life in this world, not in the United States, but in the world, is still diarrhea diseases. So it would only make sense that if we give the right formula, mother's milk, it works. We now have one protocol where we're giving human milk sugars in the ICU in people with gut failure. They come into us after multiple operations. So it's a very exciting area, and we now know it's not just the soluble fibers, but in fact the non-fermentable fibers. This is in a mouse model of sepsis. We can stop and decrease sepsis even with the non-fermentable fibers. So again, fibers I can't say enough good things about. We now know these multiple papers. You can see all 2016, 2017, decrease in all-cause mortality by a high-fiber diet, decreased diabetes, coronary heart disease, decreased cancer, et cetera, et cetera. These are very good journals, very good articles to show that. Are they safe? Bottom line is the answer is yes. We give them in our hospital to everybody. Transplant, liver transplants, everybody except bone marrow transplant. Just got to quit fighting with the ID guys. Okay. There's no data that hurts them, but there's no data that helps them. We know in liver transplant we decrease sepsis, so that was an easy no-brainer. The transplanter said, okay, we'll do it. So I think we've got to start thinking about changing the way we do things, bioecological control, rather than killing bacteria. One of our projects now, we just got a big grant from the Nike people. Phil Knight from Nike has been very benevolent. He lives in Portland. He gave us $1 billion to study cancer. He gave us $400 million to study cardiac disease. And we've got a big project now going on with us in combined University of Oregon. Not me $1 billion, he gave the university $1 billion. I just wish I had $1 billion. Anyway, we now combine, we know that if you're in certain beds in our hospital, your risk of C. diff is higher. This is a skeleton map of our hospital. You don't want to be on 14K. That's where the C. diff is. So the architects at the University of Oregon now are working with, what about remember C. diff is a spore former. Spores from C. diff can travel 9 meters. So we know now we're checking ducts, we're checking this, we're checking that all over the hospital. So that's part of that big project. So I think what we can say as I finish up here, eat a wide variety of high fiber foods that'll increase your variety, okay? Increase your diversity. Try to add fermented foods when you can. Minimize your food additives, the sweeteners, the stabilizers, the emulsifiers, the pesticides. Locate a good source of local probiotics. Don't spend a lot of money. You don't have to spend a lot of money. They're not key. Okay, daily intake with a good prebiotic whenever you can, hopefully as vegetables and fruits. Okay, remember that watch out with some of these claims. A lot of probiotic claims are pure quackery. Association to disease does not mean a causation. We got to be careful there. Remember, we got to look at the science. We can't just go with some crazy article from some crazy journal. Remember, more bacteria does not automatically mean it's better. Thank you very much.
Right, well, thank you anyway, Rich, for the introduction. So, as Rich said, I'm going to try and inform you a little bit about what goes on in your gut, and in particular, all the microbes that live in your gut and why they're so important for your health, and why, under some conditions, they can actually cause quite severe disease. So, there's been a significant shift in our understanding of what causes disease, and I think, traditionally we've always thought of it's to do with who we are, our genes, and then the things we do as we go through life. So lifestyle and what we eat and what we get exposed to in the environment. And it's those two that come together to either keep us healthy or to cause disease. But what's apparent now is that in the middle of this, and that may be involved in interpreting a lot of these things that we do and eat, are our gut microbes. And they're a direct link to our genetic material, and they can in turn influence how we react and respond to things in the environment and how they can keep us healthy or not. And really the understanding of gut microbes has really taken a fantastic leap since around 2000. This graph here shows the number of scientific articles that have been published about gut microbes. You can see they really started to take off here around 2002. And that's because of technology. So before 2002, the only way we could really identify and characterize our gut microbes was by what we could culture on a Petri dish. And since we now know that about 80% of our gut microbes can't be cultured, that really isn't a good representation of what's in our gut. But then, with the advent of gene sequencing technology, we can now identify microbes according to their genetic blueprint. And what's apparent is that different types of bacteria have a unique genetic fingerprint. So if we can identify the fingerprint, we can say whether or not they're present or absent. And this, as I said, has led to this huge explosion in this area of research. We can now identify microbes that we cannot culture. And so this has led to massive interest in gut microbes. and some of these are very recent. So the Daily Mail thinks that healthy gut bacteria might be linked to anxiety, and then we've got others that linking gut-brain connection, autism, probiotics as a means of treating diseases, and then a couple of books. This one has just come out, and those of you in the Institute will notice the significance of broccoli on the front, as the Institute is responsible for generating strains of broccoli that have lots of nutrients, good beneficial nutrients in them. But really the message here is that what we eat influences our microbes, which in turn can influence our brain function and keeping it normal. But, as always, we have to be aware of the hype. Okay, so whenever we read these articles, we need to have a couple of things in mind that allow us to determine whether or not, there's some factual basis to it or whether it's hype. And these are some of the questions that I would say you need to ask. So the obvious one is, well, so what? Do these differences they're detected, do they really matter? Are the changes a cause or a consequence of the disease? And of course we want to know how it works. What's the mechanism? So is there anything in this article that allows us to understand how it actually works? And then a lot of experiments are carried out on animals because we can't do many interventions in humans for ethical reasons. So another obvious question is, well, is a mouse a small human? No, it isn't. So we've got to bear that in mind. And then obviously we've got to think, well, is there something else they haven't looked at which could explain what they're describing? So behavior and lifestyle are two important things. So I'm going to try and sort of touch on some of these things in the rest of my talk. So this is what I'm going to cover. I think I need to introduce the gut to you. I'll talk a little bit about microbes, some interesting facts, a little bit of trivia, and then how gut microbes may play a role in determining what we eat and what the consequences of what we eat are for our health and well-being, and then how we actually might manipulate the gut microbes to improve or restore our health. So that's what I call lawn care. Right, start with, so it's the gut, mouth to the anus. It's a long tube. Here's a picture taken with an endoscope, and you can see it's not a smooth tube. It's got these ridges to it, the muscles. This is what allows food to be propelled through the gut. But it's not a smooth tube. It has lots of finger-like projections that we call villi that stick into the lumen to capture nutrients and absorb them. So the tube is quite long. It's about 9 meters from mouth to anus. And somebody has taken the trouble of trying to calculate what the surface area of all these villi are. And the outcome of that is it's probably about the size of a badminton court. So it's an incredibly large area, and it has to be large in order to take up the nutrients that are in your diet to keep you healthy. And then we also consider the process of digestion. And the gut is in fact a massive bioreactor. So we take in foods, plant material for examples, and they're broken down first of all in the small intestine here, where the small simple sugars are absorbed, and then the larger, more complex plant material that we eat in our diet passes through into the large bowel or the colon, where it's fermented. And it's fermented by the bacteria that live in the colon. And the end product of all of this is something called short-chain fatty acids, which are very important because they can provide about 5 to 15% of our daily energy requirements. In some animals, it's up to 30%. So this has to be a very efficient process to keep us alive, basically. And the enzymes that are responsible, the proteins that digest these food material and the polysaccharides, we only have about 20 genes in our whole genome that encode proteins that will break down these carbohydrates. But one bacterial species, this one in particular, Bacteroides, has 260. And you think there are 1,000 species, so that's a vast number of proteins that can digest our food. So the bacteria that live in our colon are ideally suited for processing our food and extracting the maximum level of nutrients from them. So it's a bioreactor. A little bit about the microbes. So the gut is packed full of microbes. There is no space. That endoscope image I showed you they've displaced and rinsed out all the bacteria. Normally that would be jam-packed with bacteria. Most of them are floating free in the lumen, but a large number of them actually make physical contact with the cells that line our gut. So there's actually some intimate association of these microbes with our gut. And there are two terms that you may come across that we use to describe these microbes. The microbiota, which is to describe all the microorganisms that live in the gut. And there's the microbiome, and that's all the microbes plus all their genes combined. So microbiota and microbiome, the two words you may have come across in a lot of these articles. But individually, bacteria are incredibly small. So this is the head of a pin under an electron microscope, and each of these orange dots represents one bacterial cell. So you can see that you can get lots of bacteria on the top, on the tip of a pin. They're incredibly small. But although they're small, they make up for that in their vast numbers. So we have about 10 trillion cells in our body, but we actually have 10 times that number of bacteria in our body. And so on this scale here, we have enough cells that would fill half of one of our legs. All the rest of the body will be filled up with microbes, bacteria. And then if we think about all the DNA that we have, oh, this is an interesting quote. Sorry, I forgot about this. This is an interesting, just to give you an idea of the scope and scale of the numbers here of bugs, the bacteria in our colon. So just 1 linear centimetre contains more bacteria than all the humans that have ever been born. So it's a vast number of microbes. And then the DNA elements. This is the big toe, okay, and that represents the DNA in our body that is actually ours. Okay, so everything else, more than 99% of the DNA is bacteria.
I'll take that.
So just think about that. That's actually quite amazing, really. So we are carrying around a lot of DNA, but very little of it is our own. Okay, now this is the audience participation bit. Some trivia. How much do you think all the microbes in our body weigh? Don't be shy. PhD students at the back. Come on. How much?
You might want to run.
What? Stop conferring.
Okay, ready?
So, no. Anybody else? Kilogram? Closer. 2 kilograms. 2 to 3 kilograms. A lot, right? A couple of bags of sugar. And if you put it in a volume size, about 1.5 litres.
Medic!
And there's about 1,000 different species, 1,000 different types packed in there. And this is what they need to keep them healthy. About 50 to 65 grams of these things, which are sugars, to keep them healthy. So that amount is needed every day just to keep your microbes healthy. And then you've got all the other things that you need to keep your body healthy. And so a product of all this metabolism is gas. So how much do you think we expel every day? And this is everybody, so it's not just old men and teenagers. Everybody in this room expels gas. How much do you think we expel every day?
We've got company.
How many litres? How many? 5? No, that's a bit high. Anybody else?
Yes.
1 to 4.
That's reloaded.
That's a lot. And of course, at the end of all this, we have waste. So 60% of your stool is made up of bacteria, live and dead. Okay? So that's trivia. Interesting thing. Just before you have your meal, you can run through some of these facts and figures. But they are very, very important. And we know they're very important because of animals that we can keep germ-free. So these are animals that have never been exposed to any microbes. They're sterile. And when we examine these animals, they're clearly compromised. They're deficient. So they have nutritional deficiencies. They don't grow. But interestingly, they live longer. So if you want to live longer, don't eat. That's the bottom line. They have a defective gut. So their gut is not properly formed, so it's leaky. and their immune system is very poorly developed, so they're very susceptible to infections. And in fact, if you introduce a pathogen to these animals, it can kill them very quickly because they have no protection, no immunity. And also their development is affected as well. So clearly we're already starting now to move into the gut-brain. So there are really poor animals, very sick. So the microbiota and the gut microbes are very important. So your microbiota is unique to you. It's your identity, it's like your fingerprint. Your microbes are unique to you. However, the microbes you have are shared with other family members, so there's some commonality there. And interestingly, looking at the microbiota of monozygotic and dizygotic, that's identical, non-identical twins, you know, there's no difference. So even if you're an identical twin, you'll have similar differences in your microbiota to non-identical twins. So what does that mean? Well, it means that genes are important, who you are is important, but also the nurture, the nature. The nurturing is also important in shaping the microbe. But we now know that we all have a core microbiota. So there's about 57 species of bacteria that we all share. And there are two types that predominate in all of us here. And not unsurprisingly, these are conserved because they perform important functions, such as ones here. Degradation of carbohydrates, so degrading our plant material we eat. They also provide these fatty acids that we need to keep us alive every day. And also amino acids and vitamins, which we can't produce, but our gut bacteria can. So it's not surprising there's a core that all of us need to keep us healthy, but then everything else, all the other 800 or 2,000 species are all unique to us. So where do they come from? Well, your parents, and in particular your mother. So if you think you have bad bacteria, you can blame your parents, fully justified. Okay? Now, originally it was thought that we were born sterile, but that's changing slightly. It's now evidence that we can, in fact, babies do get exposed in the womb to bacteria that the mother has, and that can be through the placenta and also via other routes. But by far the biggest source or time point at which you get exposed to microbes is soon after birth. Because if you believe we are born sterile, then the bacteria can colonize very, very quickly. So the first few months after birth, you're rapidly being colonized by bacteria. The types of bacteria depend on the delivery. So if it's vaginal delivery, then most of the microbes that will colonize the baby will come from the mother. If it's a C-section, then the bacteria actually come from the people in the operating theatre that are handling the baby. And most of those will be skin-type bacteria. And that's important because there are now evidence that links later onset of various diseases and disorders back to whether or not you were vaginally born or from a C-section and the types of microbes that initially colonize the body. Other things that will impact on the types of microbes that will colonize this infant are delivery, so is it a normal birth or is it, does it require intensive care? The age at birth is also important. Is it a full-term birth or preterm birth? And hygiene, obviously where you're born. The home versus the hospital have very different populations of microbes that can colonize the infant. And then after that, things that will impact and cause alterations in the microbes are antibiotics. And again, it depends on how many, what types, and for how long. And also, very important is nutrition, whether or not the infant's breast or bottle fed. And again, the breast milk contains lots of ingredients, including microbes, which can colonize the baby and keep them healthy. But as we go older, we get exposed to microbes from other sources and by different routes. So via the nose and lungs, we breathe microbes in, the mouth and the gut, obviously the things we eat, and through the skin. And these are the sources, so water and the food we eat will contain microbes. We have pets. If we live on a farm, we're getting exposed to different microbes from the animals that we live with. Where we live. Do we live in the country or do we live in the city? The population of microbes, again, are very different. And then the type of accommodation or the dwelling that you live in. Is it single dwelling? Is it multi-dwelling? All these people are contributing microbes that will you'll be exposed to. And then, are you indoors? Are you outdoors? Are you active? Are you inactive? Are you an Xbox fan? Or are you out playing football? These things will all expose to different types of microbes. All of these are important because beyond 3 years of age, your microbiota is pretty much set for life. So the early years of life are critical for the development of a healthy microbiota. However, there are cultural things and social things that will also impact on the types of microbes that populate us. Now here's a fact most of you probably didn't know. Okay, interesting one to experiment on. So, intimacy. And, it's across the animal kingdom. Different types of intimacy, transfer of microbes. Grooming, it's another one. Nurturing. Food sharing, right? We often sit down at the table and eat together, and we can be sharing food. It's a good way of transferring microbes. And then there's something that's slightly less, pleasant. But animals do transfer microbes to their offspring via this route by regurgitation of food and transfer of food as well as microbes. So the message here is if you have some good bacteria, you need to share it because there are some of us poor, less fortunate people, scientists for one, right? I mean, my wife is very fond of telling me I have very little culture, so maybe my culture is my bacteria. So share your good bacteria if you have them.
Energy tap is full.
Right, so we have our microbiota, we've been exposed, we've got a stable population, but it's not the end of the story. They do change. And here we've got a representation of aging. So here you can see these circles, the different colors represent different types of microbes. As we age, you can see the colors change as the populations change. And there are some differences between formula-fed and breastfed babies. Transition to solid food is a big one in terms of shifts in microbial populations. And then you can see as we age, there is also a shift as well in the population. Aging has an impact in itself. But one of the most striking impacts is through antibiotic treatment. And this slide just illustrates the impact of two antibiotics to treat Clostridium difficile, which is a severe infection that is often acquired in hospital. So as a result of the outgrowth of this bacteria, we get sick, so we're administered vancomycin or metronidazole. And what you can see is the diversity, the number of bacteria we have in our gut is drastically reduced because the antibiotics have killed them off. But it's also killed off Clostridium difficile, which is a good thing, but there's a consequence to this in that we've wiped out a lot of our good bacteria. So too many antibiotics for too long have a very profound and can be a long-lived effect on our microbiota. So antibiotics could be described as a man-made catastrophe. However, most of the antibiotics that are used in agriculture and in farm animals in particular to check infection and accelerate growth. So about 19,000 tons of antibiotics are used in agriculture every year. And of course, antibiotics get excreted by animals and humans as well, so they can contaminate streams and rivers and then get back into the food chain. And also, giving antibiotics to children has its consequences as well. So in the US, by 2 years of age, most children had at least 3 courses of antibiotics. And, there's a phenomenal number of doses of antibiotics given out in the US. And what this does is it drives bacteria to become resistant. And this is serious, okay? So this particular organism here, MRSA, now is resistant to most of the antibiotics that we have in the pharmacy. And more than 19,000 people a year in the US are killed, which is much higher than the number of people dying from AIDS. I don't know if you've just seen on the news today, but there's a UK government review group has recommended the pharmaceutical industry invest $2 billion in developing new antibiotics. There's a real need for this. But one of the causes is that we administer too many antibiotics. We take too many antibiotics. It leads to resistance. Cautionary tale. The other side of the story is that gut microbes can actually work on drugs. Rather than being affected by drugs,